Ecology. Ant mutualism. An ant guards its aphids. Ants tending aphids. Ant extracting honeydew from an aphid. Ants tending aphids and collecting honeydew secreted. A wrinkled solder beetle flies in and eats an aphid before being chased away by the ants. Some species of ants farm aphids, protecting them on the plants where they are feeding, and consuming the honeydew the aphids release from the terminations of their alimentary canals. This is a mutualistic relationship, with these dairying ants milking the aphids by stroking them with their antennae. B67. Although mutualistic, the feeding behavior of aphids is altered by ant attendance. Aphids attended by ants tend to increase the production of honeydew in smaller drops with a greater concentration of amino acids. 68. Some farming ant species gather and store the aphid eggs in their nests over the winter. In the spring, the ants carry the newly hatched aphids back to the plants. Some species of dairying ants, such as the European yellow meadow ant, Lasius flavus, 69, manage large herds of aphids that feed on roots of plants in the ant colony. Queens leaving to start a new colony take an aphid egg to found a new herd of underground aphids in the new colony. These farming ants protect the aphids by fighting off aphid predators. 67. Some bees in coniferous forests collect aphid honeydew to make forest honey. 28. An interesting variation in ant-aphid relationships involves lycaenid butterflies and myrmica ants. For example, Nipanda fusca butterflies lay eggs on plants where ants tend herds of aphids. The eggs hatch as caterpillars which feed on the aphids. The ants do not defend the aphids from the caterpillars, since the caterpillars produce a pheromone which deceives the ants into treating them like ants, and carrying the caterpillars into their nest. Once there, the ants feed the caterpillars, which in return produce honeydew for the ants. When the caterpillars reach full size, they crawl to the colony entrance and form cocoons. After 2 weeks, the adult butterflies emerge and take flight. At this point, the ants attack the butterflies, but the butterflies have a sticky wool-like substance on their wings that disables the ants' jaws, allowing the butterflies to fly away without being harmed. 70. Another ant-mimicking gall aphid, Paracletus cimiciformis areosomatinae, has evolved a complex double strategy involving 2 morphs of the same clone and tetramorium ants. Aphids of the round morph cause the ants to farm them, as with many other aphids. The flat morph aphids are aggressive mimics with a wolf in sheep's clothing strategy. They have hydrocarbons in their cuticle that mimic those of the ants, and the ants carry them into the brood chamber of the ant's nest and raise them like ant larvae. Once there, the flat morph aphids behave like predators, drinking the body fluids of ant larvae. 71. Bacterial endosymbiosis. Endosymbiosis with microorganisms is common in insects, with more than 10% of insect species relying on intracellular bacteria for their development and survival. 72. Aphids harbor a vertically transmitted, from parent to its offspring, obligate symbiosis with Buchnera aphidicola, the primary symbiont, inside specialized cells, the bacteriocytes. 73. 5 of the bacteria genes have been transferred to the aphid nucleus. 74. The original association is estimated to have occurred in a common ancestor. 280 to 160 million years ago and enabled aphids to exploit a new ecological niche, feeding on phloem sap of vascular plants. B. aphidicola provides its host with essential amino acids, which are present in low concentrations in plant sap. 75. The metabolites from endosymbionts are also excreted in honeydew. 76. The stable intracellular conditions, as well as the bottleneck effect experienced during the transmission of a few bacteria from the mother to each nymph, increase the probability of transmission of mutations and gene deletions. 77. 78. As a result, the size of the B. aphidicola genome is greatly reduced, compared to its putative ancestor. 79. Despite the apparent loss of transcription factors in the reduced genome, gene expression is highly regulated, as shown by the tenfold variation in expression levels between different genes under normal conditions. 80. Buchnera aphidicola gene transcription, although not well understood, is thought to be regulated by a small number of global transcriptional regulators and/or through nutrient supplies from the aphid host. 81. Some aphid colonies also harbor secondary or facultative, optional extra bacterial symbionts. These are vertically transmitted, and sometimes also horizontally, from one lineage to another and possibly from one species to another. 82. 83. So far, the role of only some of the secondary symbionts has been described. Regella insectivicola plays a role in defining the host plant range. 84. 85. Hamiltonella defensa provides resistance to parasitoids but only when it is in turn infected by the bacteriophage apis. 86. 87. And Serratia symbiotica prevents the deleterious effects of heat. 88. Predators. Aphids are eaten by many bird and insect predators. In a study on a farm in North Carolina, 6 species of passerine bird consumed nearly 1 million aphids per day between them, the top predators being the American goldfinch, with aphids forming 83% of its diet, and the vesper sparrow. 89. Insects that attack aphids include the adults and larvae of predatory ladybirds, hoverfly larvae, parasitic wasps, aphid midge larvae, aphid lions, the larvae of green lacewings, and arachnids such as spiders. Among ladybirds, Mysia oblongogutata is a dietary specialist which only feeds on conifer aphids, whereas Adalia bipunctata and Cossinella septempunctata are generalists, feeding on large numbers of species. The eggs are laid in batches, each female laying several hundred. Female hoverflies lay several thousand eggs. The adults feed on pollen and nectar but the larvae feed voraciously on aphids. Eupaeodes chorally adjusts the number of eggs laid to the size of the aphid colony. 90. Predators eating aphids. Ladybird larva. Hoverfly larva. The ladybird beetle Propolia. Quatorta simpunctata. Aphids are often infected by bacteria, viruses, and fungi. They are affected by the weather, such as precipitation, 91 temperature, 92, and wind. 93 Fungi that attack aphids include Neozygites freeseni, Entomophthora, Bovaria bassiana, Metarrhizum anisoplii, and entomopathogenic fungi such as Lecanicilium lecani. Aphids brush against the microscopic spores. These stick to the aphid, germinate, and penetrate the aphid's skin. The fungus grows in the aphid's hemolymph. After about 3 days, the aphid dies and the fungus releases more spores into the air. Infected aphids are covered with a woolly mass that progressively grows thicker until the aphid is obscured. Often, the visible fungus is not the one that killed the aphid, but a secondary infection. 91. Aphids can be easily killed by unfavorable weather, such as late spring freezes. 94 Excessive heat kills the symbiotic bacteria that some aphids depend on, which makes the aphids infertile. 95 Rain prevents winged aphids from dispersing, and knocks aphids off plants and thus kills them from the impact or by starvation. 91 96 97 but cannot be relied on for aphid control. 98. Anti-predator defenses. Aphids secreting defensive fluid from the cornicles. Most aphids have little protection from predators. Some species interact with plant tissues forming a gall, and abnormal swelling of plant tissue. Aphids can live inside the gall, which provides protection from predators and the elements. A number of galling aphid species are known to produce specialized, soldier, forms, sterile nymphs with defensive features which defend the gall from invasion. 28 99 100 For example, Alexander's horned aphids are a type of soldier aphid that has a hard exoskeleton and pincer-like mouthparts. 70 144 A woolly aphid, Colophina clematis, has first instar, soldier, nymphs that protect the aphid colony, killing larvae of ladybirds, hoverflies and the flower bug Anthochorus nemoralis by climbing on them and inserting their stylets. 101. Although aphids cannot fly for most of their life cycle, they can escape predators and accidental ingestion by herbivores by dropping off the plant onto the ground. 102 Other species use the soil as a permanent protection, feeding on the vascular systems of roots and remaining underground all their lives. They are often attended by ants, for the honeydew they produce and are carried from plant to plant by the ants through their tunnels. 89. Some species of aphid, known as woolly aphids, Eriosomatinae, excreta, fluffy wax coating, for protection. 28 The cabbage aphid, Brevicorine brassicae, sequesters secondary metabolites from its host, stores them and releases chemicals that produce a violent chemical reaction and strong mustard oil smell to repel predators. 103 Peptides produced by aphids, thaumatines, are thought to provide them with resistance to some fungi. 104. It was common at one time to suggest that the cornicles were the source of the honeydew, and this was even included in the shorter Oxford English Dictionary, 105, and the 2008 edition of the World Book Encyclopedia. 106 In fact, honeydew secretions are produced from the anus of the aphid. 107 While cornicles mostly produce defensive chemicals such as waxes, there also is evidence of cornicle wax attracting aphid predators in some cases. 108. Some clones of Apis craxivora are sufficiently toxic to the invasive and dominant predatory ladybird Harmonia axaritis to suppress it locally, favoring other ladybird species. The toxicity is in this case narrowly specific to the dominant predator species. 109. Parasitoids. Aphids are abundant and widespread, and serve as hosts to a large number of parasitoids, many of them being very small, circa 0.1 inches, 2.5 millimeters, long parasitoid wasps. 110 One species, Apis ruborum, for example, is host to at least 12 species of parasitoid wasps. 111 Parasitoids have been investigated intensively as biological control agents, and many are used commercially for this purpose. 112. Plant-aphid interactions. Aphids on plant host. Plants mount local and systemic defenses against aphid attack. Young leaves in some plants contain chemicals that discourage attack while the older leaves have lost this resistance, while in other plant species, resistance is acquired by older tissues and the young shoots are vulnerable. Volatile products from interplanted onions have been shown to prevent aphid attack on adjacent potato plants by encouraging the production of terpenoids, a benefit exploited in the traditional practice of companion planting, while plants neighboring infested plants showed increased root growth at the expense of the extension of aerial parts. 31 The wild potato, Solanum bertholtii, produces an aphid alarm pheromone, E-beta-farnesene, as an allomone, a pheromone to ward off attack. It effectively repels the aphid Mysis persicae at a range of up to 3 millimeters. 113 S. bertholtii and other wild potato species have a further anti-aphid defense in the form of glandular hairs which, when broken by aphids, discharge a sticky liquid that can immobilize some 30% of the aphids infesting a plant. 114. Plants exhibiting aphid damage can have a variety of symptoms, such as decreased growth rates, mottled leaves, yellowing, stunted growth, curled leaves, browning, wilting, low yields, and death. The removal of sap creates a lack of vigor in the plant, and aphid saliva is toxic to plants. Aphids frequently transmit plant viruses to their hosts, such as to potatoes, cereals, sugar beets, and citrus plants. 28 There are two types of virus transmission between plant-aphid interactions, non-circulative transmission and circulative transmission. In non-circulative transmission, the virus attaches itself to the aphid's mouthparts and is released when the aphids feed on a different plant. These non-circulatory transmitted viruses promotes rapid dispersion of the vector, or aphids. In circulative transmission, the virus is ingested and passes through the gut lining to enter the hemolymph, where it is then circulated throughout the entire body. After reaching the salivary glands, the virus is then released into the saliva upon transmission sites in plants. Circulatory transmitted viruses allows for long-term feeding by the aphids and increases the chances of being infected with the virus. 115. The green peach aphid, Mysis persicae, is a vector for more than 110 plant viruses. Cotton aphids, Aphis gossypi, often infect sugarcane, papaya and peanuts with viruses. 20. In plants which produce the phytoestrogen coumestrol, such as alfalfa, damage by aphids is linked with higher concentrations of coumestrol. 116. Aphid with honeydew, from the anus, not the cornicles. The coating of plants with honeydew can contribute to the spread of fungi which can damage plants. 117. 118. Honeydew produced by aphids has been observed to reduce the effectiveness of fungicides as well. 119. A hypothesis that insect feeding may improve plant fitness was floated in the mid-1970s by Owen and Wiegert. It was felt that the excess honeydew would nourish soil microorganisms, including nitrogen fixers. In a nitrogen-poor environment, this could provide an advantage to an infested plant over an uninfested plant. However, this does not appear to be supported by observational evidence. 120. Sociality. Some aphids show some of the traits of eusociality, joining insects such as ants, bees, and termites. However, there are differences between these sexual social insects and the clonal aphids, which are all descended from a single female parthenogenetically and share an identical genome. About 50 species of aphid, scattered among the closely related, host-alternating lineages Eriosomatinae and Hormiphidae, have some type of defensive morph. These are gall-creating species, with the colony living and feeding inside a gall that they form in the host's tissues. Among the clonal population of these aphids, there may be several distinct morphs, and this lays the foundation for a possible specialization of function, in this case, a defensive caste. The soldier morphs are mostly first and second instars, with the third instar being involved in Eriosoma moriocans and only in Smitherodes batae are adult soldiers known. The hind legs of soldiers are clawed, heavily sclerotized, and the stylets are robust, making it possible to rupture and crush small predators. 121. The larval soldiers are altruistic individuals, unable to advance to breeding adults but acting permanently in the interests of the colony. Another requirement for the development of sociality is provided by the gall, a colonial home to be defended by the soldiers. 122. The soldiers of gall-forming aphids also carry out the job of cleaning the gall. The honeydew secreted by the aphids is coated in a powdery wax to form liquid marbles. 123. That the soldiers roll out of the gall through small orifices. 100. Aphids that form closed galls use the plant's vascular system for their plumbing. The inner surfaces of the galls are highly absorbent and wastes are absorbed and carried away by the plant. 100. Interactions with humans. Pest status. About 5,000 species of aphid have been described, and of these, some 450 species have colonized food and fiber crops. As direct feeders on plant sap, they damage crops and reduce yields, but they have a greater impact by being vectors of plant viruses. The transmission of these viruses depends on the movements of aphids between different parts of a plant, between nearby plants, and further afield. In this respect, the probing behavior of an aphid tasting a host is more damaging than lengthy aphid feeding and reproduction by stay-put individuals. The movement of aphids influences the timing of virus epidemics. 124. They are major pests of greenhouse crops, and species often encountered in greenhouses include green peach aphid, Mysis persicae, cotton or melon aphid, Aphis gossypi, potato aphid, Macrosiphum euphorbiae, foxglove aphid, Olicorthum solana, and chrysanthemum aphid, Macrosiphoniella sanborni, and others, which cause leaf yellowing, distorted leaves, and plant stunting. The excreted honeydew is a growing medium for a number of fungal pathogens, including black sooty molds from the genera Capnodium, Fumigo, and scorias, which then infect leaves and inhibit growth by reducing photosynthesis. 125. Aphids, especially during large outbreaks, have been known to trigger allergic inhalant reactions in sensitive humans. 126. Dispersal can be by walking or flight, appetitive dispersal, or by migration. Winged aphids are weak flyers, lose their wings after a few days and only fly by day. Dispersal by flight is affected by the impact, air currents, gravity, precipitation, and other factors, or dispersal may be accidental, caused by the movement of plant materials, animals, farm machinery, vehicles, or aircraft. 124. Control. Parasitoid braconid wasp ovipositing in black bean aphid. Insecticide control of aphids is difficult, as they breed rapidly, so even small areas missed may enable the population to recover promptly. Aphids may occupy the undersides of leaves where spray misses them, while systemic insecticides do not move satisfactorily into flower petals. Finally, some aphid species are resistant to common insecticide classes including carbamates, organophosphates, and pyrethroids. 127. For small backyard infestations, spraying plants thoroughly with a strong water jet every few days may be sufficient protection. An insecticidal soap solution can be an effective household remedy to control aphids, but it only kills aphids on contact and has no residual effect. Soap spray may damage plants, especially at higher concentrations or at temperatures above 32 degrees Celsius, 90 degrees Fahrenheit. Some plant species are sensitive to soap sprays. 112, 128, 129. Green peach aphid, Mysis persicae, killed by the fungus Pandora neoaphidus, entomophthorals. Aphid populations can be sampled using yellow pan or moric traps. These are yellow containers with water that attract aphids. 130. Aphids respond positively to green, and their attraction to yellow may not be a true color preference but related to brightness. Their visual receptors peak in sensitivity from 440 to 480 nanometers and are insensitive in the red region. Moric found that aphids avoided landing on white coverings placed on soil and were repelled even more by shiny aluminium surfaces. 131. Integrated pest management of various species of aphids can be achieved using biological insecticides based on fungi such as Lecanicilium lecani, Beauveria bassiana or Isaria fumoserosi. 132. Fungi are the main pathogens of aphids. Entomophthoros can quickly cut aphid numbers in nature. 133. Aphids may also be controlled by the release of natural enemies, in particular lady beetles and parasitoid wasps. However, since adult lady beetles tend to fly away within 48 hours after release, without laying eggs, repeated applications of large numbers of lady beetles are needed to be effective. For example, one large, heavily infested rose bush may take 2 applications of 1,500 beetles each. 134. The ability to produce allomones such as farnesene to repel and disperse aphids and to attract their predators has been experimentally transferred to transgenic Arabidopsis thaliana plants using an E-beta-F synthase gene in the hope that the approach could protect transgenic crops. 135. E-beta farnesene has however found to be ineffective in crop situations, although stable or synthetic forms help improve the effectiveness of control using fungal spores and insecticides through increased uptake caused by movements of aphids. 136. Insects portal. In human culture, aphids are familiar to farmers and gardeners, mainly as pests. Peter Marin and Richard Mabey record that Gilbert White described an invading army of black aphids that arrived in his village of Selborne, Hampshire, England, in August 1774 in great clouds, covering every plant, while in the unusually hot summer of 1783, White found that honeydew was so abundant as to deface and destroy the beauties of my garden, though he thought the aphids were consuming rather than producing it. 137. Infestation of the Chinese sumac, Rus chinensis, by Chinese sumac aphids, Schlectendalia chinensis, can create Chinese galls, which are valued as a commercial product. A's. Gala chinensis. They are used in traditional Chinese medicine to treat coughs, diarrhea, night sweats, dysentery and to stop intestinal and uterine bleeding. Chinese galls are also an important source of tannins. 28. Stridulatory sound production and its function in females of the cicada subsaltria yangi. Editor. Alex Cordova. Abstract. Acoustic behavior plays a crucial role in many aspects of cicada biology, such as reproduction and intrasexual competition. Although female sound production has been reported in some cicada species, acoustic behavior of female cicadas has received little attention. In Cicada subsaltria yangi, the females possess a pair of unusually well-developed stridulatory organs. Here, sound production and its function in females of this remarkable cicada species were investigated. We revealed that the females could produce sounds by stridulatory mechanism during pair formation, and the sounds were able to elicit both acoustic and phonotactic responses from males. In addition, the forewings would strike the body during performing stridulatory sound-producing movements, which generated impact sounds. Acoustic playback experiments indicated that the impact sounds played no role in the behavioral context of pair formation. This study provides the first experimental evidence that females of a cicada species can generate sounds by stridulatory mechanism. We anticipate that our results will promote acoustic studies on females of other cicada species which also possess stridulatory system. Introduction. Acoustic signaling is a widespread form of communication and occurs not only in vertebrates, but also in arthropods and even in plants 1 to 4. Among animals, acoustic communication is used in different behavioral contexts, such as intrasexual competition, intersexual interactions, and territorial defense 2, 5, 6. For example, sound-based communication plays a vital role in reproduction in a diverse range of taxa, e.g., frogs and insects, in which acoustic advertisement is generally the domain of males 2, 6. The number and kind of communicative signals in the repertoire of an animal species depend on the complexity of its life activities 7. Different acoustic properties of communication signals potentially encode different kinds of biologically significant information such as sex, dominance status and physical condition 8. Sound-producing behavior has evolved in diverse arachnid and insect groups. Many species of spiders can produce sounds by stridulatory mechanism, and 12 different types of stridulatory apparatus are known in spiders 9 to 11. Acoustic signals of spiders are used in intersexual communication, intrasexual agonistic interactions, or to reinforce other defensive signals showed towards potential predators 12 to 14. In the insecta, sound-producing structures may involve almost any part of the insect's exoskeleton 15, and the main sound-producing mechanisms are vibration, percussion, stridulation, clicking mechanism, and air expulsion 16, 17. Among insects, cicadas are well known for their timbal sound-producing mechanism in males 18. The timbal organ is essentially composed of a ribbed membrane at the base of the abdomen and an attached muscle, and sounds are generated when the timbal muscle activity deforms the stiff membrane 19 to 21. Male cicadas emit different types of acoustic signals in different behavioral contexts in order to gain benefits such as attracting conspecific females and deterring predators 18, 22 to 25. Sound production is generally thought to be restricted to male cicadas, and most previous studies have focused on acoustic behavior of the males. However, in some cicada species, females are able to generate sounds, functioning in intersexual communication, by means of wing flicking 26 to 30. Females in these cicadas usually do not have any specialized sound-producing structures except that in some species the forewing costa is distinctly angled 31, 32. What is interesting and unusual is that females of the cicada subsaltria yangi chen possess a pair of stridulatory organs 3-3. The stridulatory organ is a phylum scraper system similar to that found in crickets, katydids and grasshoppers 32 to 34. S. yangi is a medium-sized cicada with adult body length of 28.0-33.2 mm, and is the only known cicada species of the subfamily Teddigodinae in China 33, 35. In the present study, we conducted scraper ablation experiments to illustrate how females of S. yangi produce sounds. Furthermore, acoustic playback experiments were carried out to explore how males respond to sounds produced by S. yangi females, and how different components of the female sounds function in the behavioral context of pair formation. Materials and methods ethics statement. No specific permits were required for this study. This study did not involve endangered or protected species, and the cicada subsaltria yangi used in the present study was not included in the list of protected animals in China. Study site and species. The study site, Chunshugo Valley 38 degrees 33.699 N, 105 degrees 55.217 feet E, where the cicada subsaltria yangi is particularly abundant, Figure 1, is located in the Helingshan National Nature Reserve, Ningxia Wei Nationality Autonomous Region, China. This cicada population occurs at elevations between 1400 m and 1600 m. Investigations were performed between 6 and 26 June 2014. Adults of this cicada species feed mainly on Ephedra lepidosperma, Ephedricea, a medicinal plant used in traditional Chinese medicine. Voucher specimens are deposited in the scientific collection of the Entomological Museum, Northwest A&F University, NWAFU, China. Fig. 1. A female of subsaltria yangi. Open in a new tab. Scale bar, 1 cm. Morphology of the stridulatory organ. We examined the morphology of the scraper and the file using an Olympus SCX-10 stereo microscope, Olympus Corporation, Tokyo, Japan. The number of ridges of the file was counted. Micrographs were captured with a Rediga 2000R digital camera, Q imaging, Canada, mounted on a Nikon SMZ1500 stereoscopic zoom microscope, Nikon Corporation, Tokyo, Japan, and then 80 sequential shots at different focal depths were processed using the Auto Montage Pro software to generate a single composite image. The morphology of the file and the scraper were also examined using a scanning electron microscope, S3400N, Hitachi, Tokyo, Japan. Sound recording and analysis. The sounds produced by S. yangi females were recorded using a linear PCM recorder with stereo microphones, PCMD50, Sony, China, frequency range 20-20,000 Hz and a 44.1 kHz, 16-bit sampling resolution. The sounds were recorded in WAV file format, and the sounds recorded on the left channel of the recorder were used for acoustic analysis. Acoustic analysis was conducted using the Raven Pro 1.4, the Cornell Lab of Ornithology, Ithaca, NY, USA, and the C-Wave Package 36, a custom-made library of the R software platform 37. Scraper ablation experiments. Females of S. yangi produced sounds in response to acoustically advertising males. Behavioral observations on the sound-producing behavior of S. yangi females indicated that sounds were emitted during the rapid upward and downward movements of the forewings, S1 video. The forewing movements might cause interaction between the scraper and the file, Figure 2a, so the sounds were probably generated by the stridulatory mechanism. However, similar to the sound-producing mechanism of the wingbanger cicada Platypedia putnami, the sounds might be produced by banging the tegmina against the body 34. Therefore, it is difficult to explicitly determine the underlying mechanism of sound production in S. yangi females. To determine the role of the stridulatory organs in sound production, we conducted scraper ablation experiments in which sounds recorded from females with and without stridulatory scrapers were analyzed and compared. Fig. 2. Scraper ablation experiment. Open in a new tab. A. The red arrow indicates one of the paired stridulatory organs of a female. B. The red arrow indicates that the scraper of the stridulatory organ was removed. The captured females were kept on their host plants covered with gauze netting until testing. Scraper ablation experiments were conducted only with mature and unmated females, because our previous fieldwork had demonstrated that only unmated mature females of this species were acoustically responding to advertising males, which is the same to periodical cicadas 28. The captured females were tested individually. A female was placed in a small cage, 0.25 times 0.25 times 0.50 meters, in which the insect behaved normally according to behavioral observations. The sounds emitted by the caged female were recorded. The paired scrapers of the female were then carefully removed with surgical scissors, Figure 2a, b. When the female performed normal sound-producing movements, i.e., upward and downward movements of the forewings, the sounds emitted by the female were recorded second time. A total of 15 females were used in scraper ablation experiments. All experiments were carried out between 9 a.m. and 3 p.m., a period corresponding to the peak singing activity of this cicada. During the experiments, the ambient temperature ranged from 29 to 34 degrees Celsius. Male responses to female sounds. Behavioral observations suggested that the sounds produced by S. yangi females functioned in the behavioral context of pair formation. To determine whether the sounds produced by subsaltria females elicit acoustic and phonotactic responses from conspecific males, we conducted acoustic playback experiments in the field. A high-quality sound recording, i.e., having high amplitude relative to background noise and no overlap with other sounds, from a female of S. yangi was selected for playback experiments. Playbacks were conducted using a Sony PCMD50 linear PCM recorder and a Magic Q2 loudspeaker, frequency response, 150-20,000 Hz. A digital sound level meter, Benetech GM1357. Fast response, a weighting, was used to measure sound pressure levels. The peak output intensity of the loudspeaker was adjusted to 60 decibels SPL measured at 50 centimeters from the loudspeaker. This SPL is within the range of natural variation recorded in the cicada population range, 55.4-65.8 decibels SPL, mean plus or minus SD equals 60.5 plus or minus 3.2 decibels SPL, N equals 19. We performed experiments between 9 a.m. and 3 p.m. Vegetation in the dry habitat occupied by S. yangi consists primarily of drought-tolerant dwarf shrubs and herbaceous plants, generally not exceeding 1 meter in height. We tested whether the female sounds elicit phonotactic responses from conspecific males in the field. A total of 25 males were used for this experiment. We placed the loudspeaker on the ground and about 3 meters from an acoustically advertising male. Then, the acoustic stimulus was presented from the loudspeaker. The repetition rate of the sounds was 82 sounds per minute. During the playback period, a phonotactic response was noted if the male flying towards and landing within 50 centimeters of the loudspeaker. A no response was noted if the male flying away or remaining still after 3 minutes of the playback.